Hey, everybody, welcome back to yet another Journal Club brought to you by Leaf and Lifespan.io. I'm Dr. Oliver Medvedik, presenting as usual from New York City. We have yet another journal article, and if you want to check out the kind of the overview of this paper, this one is written up on our blog, which is available on our website. I forgot who wrote the blog article on this particular paper that involves the dream complex and DNA repair. But do check out our news articles on our website because we do cover a lot of the papers that we cover here in depth. So if you want to basically get a sort of a bite-sized overview, do check it out so you can kind of get a bird's eye view of it. So welcome back. And what paper are we doing right now? Let me take a look. Oh, this one here. Okay, let me start reading it. All right. I read it on the flight here. I was out in Colorado. I am back. Read it a couple of times. It is a C. elegans paper, mostly C. elegans, but then it dips into mice and human cells at the end. So let's take a gander here. Share. All right. How's that looking? The dream complex functions as conserved master regulator of somatic DNA repair capacities. So what's this all about in general? Well, DNA repair is really, really important for the maintenance of lifespan. And it's important for aging if it breaks down. So it's kind of one of those mechanisms that's required to kind of keep entropy at bay. It's the way I see it. So in metazoans, you've got all sorts of different types of cells. You know, you've got stem cells, you've got terminally differentiated cells, but you also have somatic cells and germline cells, right? Germline cells are the ones that produce sperm and egg, propagate the species, and then you have somatic cells. And it turns out that somatic cells have way less capability of DNA repair than germline cells. So the questions the authors had here is, well, can we boost this DNA repair capacity of somatic cells and improve the resilience of these worms, and by extension, other cells as well? So what is this dream complex? And the authors, it's, if you look at review papers, it's considered by some folks as the conserved master regulator of somatic DNA repair capacities. It acts as well, they're looking at it as a repressor here, but I think it can also activate work as an activator. I might be mistaken, but it turns on a lot of genes. So let me just stop sharing here and pull up a review paper here. I'm not going to go through the review paper, but I'm gonna just show a figure. See how this looks here. How's that look? You can see a flowchart on the left. There we go. So this dream complex, don't exactly know what it stands for. There's a lot of different proteins there. So you could see this glom of different proteins. So years ago, people were trying to figure out how P53 initiates cell cycle arrest and turns on repair and so on and so forth in lots of different cells. And P53 doesn't directly repress a lot of genes, but does it indirectly through this dream complex. I think when it comes to P53's activity as a repressor, this is from this review paper by, I think it's England, Kurt England. So P53, is one of the links in sensing DNA damage. It turns on tumor suppressors such as P21. P21 then activates things downstream. One of the things it activates is the dream complex. It can do it indirectly through a number of other different proteins, which the authors point out later in this paper. And then this complex basically does a lot of things. It represses a lot of genes. And the consequence of this repression is it essentially inhibits mitosis. So it basically, slows down cell cycle arrest. And it also, interestingly, turns down a lot of genes, downregulates a lot of genes in DNA repair as well. So one caveat I had, I'm going to pause this when I was reading this paper, is that What these authors are doing in this paper is making mutants in worms of various components of this dream complex. And the components there, I'm going to share this again. These mutations that they're working on, these are components in this dream complex that actually contact these DNA regions where it actually acts as a repressor. So there's a lot of obviously complicated stuff happening here. So we don't have really time to go into it, but here's a bunch of components that have homologs or analogs in worm. I believe this paper is covering subunits that are in mammalian cells. So E2F4, 5DP. So these are some of these elements, CHR, I believe, cyclin homology region elements, E2F regions, so Lin54 here. So you have this, contacting happening here. And depending on how this complex is activated, it can contact different sites to essentially repress transcription. So I'm going to stop sharing here for a while. So that's kind of an overview of the dream complex. So those blobs at the bottom are some of the components, I believe, that the authors are mutating in the analogous dream complex in worms, and then basically disrupting its repressor function in worms in various stages of development. So through the larval stage of development, all the way through the adult stage of development, and then seeing what happens when you stress the animal. Okay. Oops. So let me pull out my paper here. Let's go to the actual paper. Okay. So this is by a number of different labs. I believe some of them are in Germany. Arturo Ujarabal, Dueso et al. The labs are located at Cologne, Germany, Institute for Genome Stability, apropos to this paper, Institute for Molecular Biology Biotechnology. Where's this? University of Crete, Greece, another place in Germany. So quite an international consortium here. Okay, so let's take a look at some of the first things that they do. So some of the first things that they do is to see what genes are basically being regulated by this dream complex here. So let me get my notes here because I don't really work on C. elegans. So let's go to figure 1. So DDR gene promoters carry the CD-CHR dream binding motif. So they're looking at, so the first thing they do is they basically do an analysis of kind of all the promoter regions in the genome of C. elegans. So DNA damage response gene or promoters, I believe. Yeah, DNA damage, response promoter regions. So they're looking for these elements that I mentioned earlier that are these binding motifs for the dream complex, and they're seeing where they're enriched, right? So so here are these various motifs. So, and you've got C. elegans well characterized already, its entire genome, so you could see what categories of genes have these promoter elements. And this is percentage of, I think, all genes, so background genes, and percentage of DDR genes. So whenever I hear DDR, my girlfriend loves Dance Dance Revolution. So it's not Dance Dance Revolution genes, it's DNA damage response genes, I believe. Okay, enough of the jokes. So 46% versus 28% of background genes have these DPL1 or E2F. I believe that's the homolog in mammals, and EFL1 and Lin15B. So these elements are enriched. So this is basically full change, how much of an enrichment. So when they search through in these CD-CHR elements, again, that I mentioned earlier. So so you have a significant enrichment of these elements that, that the dream complex binds to, that falls into, more towards the DNA damage response genes, suggesting that if you disrupt this repressing function, you could be turning on these genes as well. More so the DNA damage response genes than other genes, but still, it's quite a lot of overlap there. So this complex, when they do these studies, they get some paradoxical results that I noticed in Figure 1, and they mention it in the paper. So they do mention, that DNA repair is essential, for improved longevity and lifespan. And it is in this case here, when you subject these worms to a stress of, let's see, I think this is ionizing, this is UV radiation, so ionizing ultraviolet radiation. They try a whole bunch of other genetic insults later. They use ionizing radiation and anti-tumor agents such as cisplatin and mutagens such as methylmethane sulfonate, but we'll get to that later. Yeah, Micah.
Just a quick question that if this answer is long, then feel free to skip it. How do you read those motif things, like the little DNA sequence there? Is there just like a one-sentence answer for how you're supposed to read that?
Yeah, you mean this funny like circus alphabet here with
exactly the circus.
Yeah, so yeah, it's kind of silly, isn't it? So basically, if you read it from one end to the other, which is 5' to 3', this is the they could have just put numbers or something there, but this is the size of the letter is supposed to represent what proportion of the entire sequence, like if you sequence it, what do you find in this location? What's the canonical sequence, right? So if I look at this point here, it's just a T. There's nothing else. So you'll only find a T here. You only find a T here. Here you only find C's and T's in equal percentages, and here you only find A's. And here you'll find, like, I can't even see, but all four of them, I guess, at this location. Does that make sense?
Gotcha. So basically, this is just a way of showing across a given organism, across many cells, you might see many mutations, and this is kind of showing the average of what the sequence is across all.
So it's basically the canonical sequence. So they do this a lot in promoters, and basically if you do this with a whole bunch of other promoters, you'll notice that if you do take the canonical sequence, let's say for this motif, and let's say you were to generate a sequence where you had, the T, C, 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 T, T, and these T's, this would be a very strong, strong sequence. If you were to just put something else in those places, then the binding would get weaker and weaker and weaker as you drift further away from the canonical sequence. And for some promoters, interestingly, because the way regulation works for promoters, a lot of promoters, at least in bacteria that I'm aware of, don't have, like, if you were to analyze like 1,000 promoters and you would get the canonical sequence, I think it's very rare to find the canonical sequence because it would make it a very strong promoter and very difficult to regulate by other sequences that are around it. So then evolution tends to kind of drift it away, and then other sequences can basically act as activator sites and so on and so forth. But anyway, that's what it's supposed to represent. This kind of wacky, way to show it. Yeah.
Would it be a stretch to say that the sections that are equal proportion ACGT are kind of inconsequential in the function of that protein to some extent?
Yeah, to that sequence, it could be what might be happening again. You'd have to look at the actual structure of how that protein blob binds and what amino acids bind. But let's say, as an example, if you look at like the -35 and -10 site, which is like the promoter of most bacteria, parts of those sequences, you have this protein called a sigma factor that binds to it, and you can imagine it kind of making contacts. So I would guess that the ones that are more variable, the amino acids on that protein glob are probably not directly contacting it, and and you'll notice also that these motifs have different lengths, right? So the length is also in many cases important, because there's a conserved size to what's contacting it. So if you were to put some nonsense here that makes this motif longer, it might not function, because the part that contacts this CGD, albeit weakly, and this series of Gs would be way out here, and the protein, well, the protein can't stretch itself, right? So sometimes even though the sequence here is variable, the length very well may not be variable. Yeah,
functioning like a spacer, basically.
Exactly. Yeah. So basically, so that's kind of it. The sequences are important in some locations where the amino acids are contacting, and also the space is important. So basically to have everything aligned, essentially. So yeah. So this is, so they do a lot of these other studies later in this paper where they look at these sequences and show that there's an enrichment of them. So they notice that, and they look at these mutations here in worms. So they have a Lin52. I don't know what N77 somebody who's a worm guy can probably tell, or a worm gal. It's probably some point mutation that corresponds to nucleotide 771, but I could be totally wrong. DPL1, ELF1, Lin35. So these, I believe, are all components of the dream complex that's found in worms. So that's why they refer to it as master regulator, because I think it was first identified in mammals and humans, but now it's found in a whole bunch of other things that repress proteins in response to p53. So larval stages. I don't know much about worms, but there's, I believe, 4 larval stages here. So you have an egg, it hatches, it goes through, I guess, several stages of development. Worms have roughly, I think they mentioned here, 995 cells, and maybe a bit more, 1,000 plus, if they're, depending on the sex they are. And most of these cells are somatic, except for a small subset that are germline. And most of those somatic cells, I think most, if not all, are post-mitotic as well. So these mutations here are supposed to disrupt the repressor complex. So by disrupting this repressor complex, you would expect the authors would hypothesize that you have increased capabilities of DNA repair. And they look at it at different stages. So L1 is the larval 1 stage, L2, L3, L4A. And basically, the gist here is that you want the cells to go to L4A. So this is adult. You want them to go all the way through. So as they're basically growing and dividing, they're getting to adulthood. So they're looking at now at the larval stage. So all the way from L1 to L4A, and they're able to, worm geneticists can look at the morphology and tell you whether the worms are in either of these stages, right? So they're going through their version of babyhood, childhood, puberty, and adulthood, right? So 0 is, 0 millijoules per square centimeter of UV light, and then they blast it with 40 and 60. So I guess these are standard, amounts that you could zap these cells with. What are we looking at for D? I'm just going to jump ahead here because it's just just checking if there's a difference. I mean, there's a difference in how many joules there are being blasted with. Anyway, so let's get back here. Obviously with 0, there's no difference. Everything kind of more or less gets to adulthood, except for these weird mutants, these Lin35. So they mention here that some of these mutants affect development, which is this Lin35. Other ones don't. So this Lin52, which they then focus on for the rest of the paper because it seems to have the least kind of severe phenotypes of these worms, and we'll take a look at the lifespan data down below. But the bottom line here is that if you look at as the UV intensity increases, you can see in wild type a lot of the cells get stuck at earlier larval stages, and these colored bar graphs here tell you what percentage are in a certain larval stage. So you can see wild type, the more you go up, less can make it into adulthood. Somebody have a question? Yeah, Micah.
When you say they're stuck in those stages, that means like they functionally die. This isn't like the desirable stuck at youth. This is the undesirable stuck. Like you can't even develop into a functioning organism. Is that correct?
Yeah, I believe so. See, let me take a look at here. See, where did they mention that there? It's undesirable. Yeah, so they're not eternally youthful here. That's okay. So they are. Just trying to see here where they mention. I'll come back to that, but I'm going to assume that they are messed up, and that they will croak at that stage.
They'll die due to lack of function or lack of ability to move forward or whatever. Yeah.
Yes. So subjecting them to heavy dosages of that. So they basically terminate at that point. So the trend basically is, so with all these little stars that show you statistical significance, is that compared to wild type, which is here and here, so going up in intensity, more of them, if you have these, if you have even in this one here, which is initially messed up, you basically, more of them are able to get to a later larval stage without dying if you subject them to higher UV light versus the wild type. So basically, it confers UV resistance in this case. And for D, see what's D here. I got to take a look at this figure legend here because it's very similar. The only thing is that the joules seem to be a little higher. Let's see if there's anything else significant here. But you can ask a question while I'm scrolling down into the figure legend.
I'm still a little confused on this chart. So I would assume that the desired outcome is that the 60 millijoules per centimeter squared would look exactly like the zero, right? Like that's what a perfect solution or perfect community. Mm-hmm. And so it feels like the Lin 52 is one of the better ones. Like they dropped that one because it's worse, but like those bars look
no they don't drop that one. They stick with Lin 52.
Oh, okay. I just misunderstood you then. Okay, so Lin 52 is the good one because the 40 and 60 bars look very similar to the 0 bar, and that's what we want.
Yes, right? Yes. Okay. So basically, the Lin 52 seem to have the best kind of phenotype, and they stick with it. Let's see, what are they doing in what's so different? I mean, figure D is more of the same. Let's see, it's higher UV radiation. You get even better results with Lin 52, 50, 70. You get more wild type, adult, coming through. Same thing with DPN1, and so they do a double mutant here. So let's see, Lin 52, DPL1. Let's see, do we have a? Oh,
pretty promising.
Yeah, pretty promising too. So they have a double mutant here, so it's also having doesn't seem to be an additive effect, but I guess Lin 52 is probably pulling more of its weight for whatever reason. The interesting thing that I find, though, is that, these worms are more robust and they seem to be able to withstand DNA damage. Initially here with UV, later we'll see with other DNA damaging agents. But for most of them, it seems that the lifespan is affected when you don't basically subject them to UV radiation. So what we're looking at here in E is, let's see, what's the 400 represent here? God, they make these things so small. So let's see, where's CD? 400 millijoules per, so a lot more, 400 millijoules per square centimeter. They mentioned in the paper that these worms are really usually robust to DNA damaging agents. So I guess to get an effect for lifespan, they really have to crank up the UV light.
Could also be that just they're doing so adult worms versus larval worms, and larval worm is simply smaller and can't handle the load as much, maybe.
Yeah, good call on that. That might be the the other point to it. So let's take a look at Lin52. That was doing the best. So wild type 0 is not being, zapped. So wild type 0 is this black line, or is it black line here, dashed line out here. Lin52, it looks initially that maybe a little less, but if they do the statistics, they say it's not significant. It actually does live, the maximal longer. So we'll accept that it's not significant. The bottom line is that, knocking out, so number one, knocking out this repressor function using these types of mutants, at least for Lin52, doesn't make the worms live shorter significantly, but it doesn't make them live longer. So it's not, you would expect boosting repair activity to make things live longer. And then kind of more paradoxically, so I'll get to when they zap. So when they zap the cells, that's when you see the more of an effect. Wild type at 400 millijoules, you see a significant decrease in lifespan right here, this black line. But not as much as a drop with Lin52, and you see the same thing when we look at other mutants as well. However, when you look at other mutants, you do see a significant decrease in lifespan at 0. So meaning it's not being subjected to any UV radiation, but you get more of a buffering against UV damage. So my thoughts on this, I think they mention it very briefly in the paper, and they say that, well, it could be what they mention here. Worms and mutations significantly outlive upon DNA damage, despite the fact that some are short-lived without irradiation, and that it might be that under unperturbed conditions, they might be connected to their previously described roles in contributing to developmental processes. Yeah, I mean, it just seems that at least in this case here, that they seem to go through, I believe, development pretty well at 0, at least ELF1 and DPL1. Maybe not Lin35. That one seems to be a little, you could say that its development is a little screwed up. So my thought on this is, I'm just going to toss this out there, is you would expect increasing DNA repair capacities. And I haven't looked at any recent C. elegans papers, and I'm sure a lot of you in the audience have looked at them. So there's other ways that you can upregulate DNA damage responses. Other ways are just basically upregulating or overexpressing DNA repair enzymes. I'm not sure what effect that has on worm lifespan. I would assume that it would increase it. It could possibly be that, this dream complex regulates hundreds and hundreds of genes. So it could be that you're seeing this, you're seeing some buffering against damage when you actually induce damage, but there are side effects that basically kind of abrogate its long-term survival, and it might be kind of too crude of a tactic, perhaps, to make these mutations in the dream complex and expect a lifespan extension. So there might be more subtle ways of doing this and upregulating the DNA damage response genes without making direct mutations in the dream complex. That's my 2 cents. If anybody else has any comments, and this is basically a bar graph that summarizes this decreased mean lifespan upon UV treatment, right? And so less decreased lifespan after UV treatment. But you do have a decreased lifespan sort of basally without actually inducing UV irradiation. So that's kind of the paradox for me, right from the get-go in this paper. Any thoughts, comments?
It is kind of curious why nature would go to so much trouble to disable DNA repair unless it's a side effect of some other function of this complex that we don't know about yet.
Yeah, so let's see. So this is from this review paper. Yeah, this dream complex does a lot. So that's the issue. Oops, let's see if there's a summary here. Well, these are all repair functions that regulates, but right here. So it doesn't just regulate DNA, damage. It's cilia assembly, G2M checkpoint, transcription. Well, also basically cell cycle regulation, right? So the other paradox that I had here was that these cells do, and they'll later show in the paper, do have less apoptosis when you dysregulate the dream complex. And these cells go further through development, right? They have increased lifespan. I don't know much about worm biology, so I don't know if worms get cancer. So if you do have dysregulation of the gene complex in that review paper I pulled up by Kurt England et al., I believe that if you have defects in the p53 dream complex, you have basically a general loss of checkpoint control. So actually deregulation can also promote chromosome instability, they say, and aneuploidy of cancer cells. So the cells will divide better, but it's kind of an aberrant division, right? So I think it does a lot of multitasking. Yeah.
Do we know if the dream complex is up or down regulated with age? Like, is there a correlation between its regulation and age?
Yeah, it's a good question. It is. Let's see what it says inside this. It is perturbed in certain diseases. Where is it perturbed in?
Is one of those diseases aging?
Yeah. Is disease and is aging a disease, right? Well, it is perturbed in Alzheimer's disease, which is a disease of aging. And they mention it's I think it is upregulated. They mentioned there in is it upregulating Cockayne syndrome, which is an accelerated aging phenotype. It's later in the paper. Let's see where they mention this. Could be later in the later when they basically look at other.
It's possible just simply no one's looked, I guess. Is a dream complex relatively new-ish discovery, or is it something that's been around for 100 years and everybody's known about?
It's kind of. I think it's new-ish. I'm not. Yeah, I mean, P53 has been around for the proverbial 100 years, and people didn't know. People didn't know how P53 was repressing a lot of these genes directly, and it turns out that later on that they found that the dream complex was doing this. So that came way after P53. So yeah,
it's possible that just no one's looked to see is there a correlation between aging and dream complex regulation. That would be I think the next thing that I would look into after this paper would be that, figure out, is this age correlated? If it is, then it gives a kind of strong evidence that maybe this is something we should try to repress or knock out. And if it's not age correlated, then I'd say, probably something else, and maybe we should leave it alone. Like, it probably has those good effects that we want.
Yeah. The only caveat I would say to that is just because it does so many things.
Yeah, certainly.
If misregulation of the dream complex, if deregulating dream target genes promotes chromosomal instability, then you're going from less aging to more cancer, right? So I think that's a there's kind of a lot of stuff happening under the hood here. And some of the components of this dream complex kind of vary, so it's not just like one dream complex. It basically can adapt other protein components to turn on and switch off other genes. So I think, there has to be a little bit of, just like the OSK factors, a little bit of delicacy there as to how you're basically controlling it. That's about it. Yeah. But yeah, definitely more further research is warranted. What do they look at in F? So they look at number of eggs. So same thing. So UV irradiation, and looking at eggs laid and eggs hatched. So this pertains to the germline. of the cells, and what they basically predict here is that these mutations won't have as much of an effect on the germline as it has on the resilience of the somatic cells, just because that this dream complex is already supposedly upregulated. I don't think they quantified here, but upregulated robustly, downregulated robustly in the germline. So you have basically more repair capacities taking place. And what do they look at here? So they look at number of eggs laid. So this is wild type. So this is the Lin52 mutation. XPC1 is DNA repair enzyme that's used in.
Presumably, more eggs laid is better.
More eggs laid is better, and more eggs hatched is better as well. Yeah. So let's see. I'm just trying to look at for XPC1 mutant here. I think it's the homolog of xeroderma pigmentosa, which is a mutant in humans, and I believe I think it's involved in nucleotide excision repair. I gotta double check that. Yeah.
So this one's interesting because just at a glance, maybe it's not significant, but at a glance, it looks like this is the opposite result, where when they're not exposed to the UV light, the Lin52 improves, just like compared to the wild type. But then once you expose the UV, Lin52 hurts. Like the wild type mouse is laying more eggs.
I noticed that here. Let's see. These significant bars are here. I'm not sure. Is this supposed to these are big, huge bars here. Maybe
again, just at a glance.
I see what you're pointing out there. I'm not sure if let's see where's the. I like it when figure legends used to be on the same page. Here it is, 3 pages down. All right. Okay, I'm going to assume that that's not statistically significant, although it kind of looks like it, right? So let's see. So yeah, wild type Lin52, not really much of an effect. XPC1, which is if you knock out, I think this is one of the DNA repair enzymes that is supposed to play a major role here during in the germline. If you knock out Lin52, it doesn't, you block the effect of basically, of its activity. So it's dependent on this DNA repair enzyme that's regulated by Lin52. Yeah.
But only if you expose it to UV light. Like if you don't expose it to UV light, it does neither XPC nor Lin52 seem to have any negative effects. Maybe a
yes.
One. Then you're exposed to UV light, which these things are supposed to be protecting against, and now the worms cannot reproduce, which is bizarre and backwards. It's like the UV light is. I don't even know how to explain that. That just seems very bizarre to me.
Well, let's take a look. I mean, number of eggs laid is the same. So if you don't expose it to UV light, if you expose it to UV light, you have let's see 50. Let's jump to 100 millijoules here. So let's get roughly a 50% decrease. So assuming that you already have as much de-repression or deregulation taking place here, then having a Lin52 mutation shouldn't have any effect. So the Lin52 and a wild type should look identical, which they do. If you knock out XPC1, which is one of the enzymes that is downstream, you basically have you basically destroy the germline essentially. And knocking out Lin52 is not going to have any effect because this downstream component is already knocked out.
Sorry, my internet dropped out, so I maybe missed it, but I feel like that doesn't answer the question of when you knock out XPC and you don't expose it to UV light, no negative effects, maybe a positive effect even. It's only if you knock it out and expose to UV that you get a negative effect. It's like the UV, the damage caused by the UV is magnified when XPC is missing. Like especially in that last chart, right? Like they're still able to reproduce with XPC when exposed to large amounts of UV. But when you knock out XPC, now they can't reproduce at all. But only when UV is present. If you remove the UV, then they can reproduce just fine without XPC. Like that's the part that's just super weird for me.
Yes, well, because there's dozens and dozens of DNA repair enzymes, and some of them are required if you basically go above a certain threshold. So I don't think you'd be able to. I think they were mentioning later that worms are really resilient to internal genotoxic stress. And the only way that they could see these effects if they basically subject them to an externally high dose that kind of overwhelms their DNA repair capacities. So that's their explanation. I'm not a worm guy, but only if you're subjecting these worms to an external stress that's over and above and beyond what all of these kind of intrinsic DNA repair capacities and all these enzymes that are found in the genome-wide nucleotide excision repair, homology, recombination, NHEJ. There's a whole bunch of these other pathways. So if you knock out a lot of them, you won't see necessarily an effect unless you knock out all of them. And this isn't XBC1, isn't all of them, but you'll only see this effect if you sort of overwhelm the system beyond a certain threshold externally. That's their explanation.
Yeah, maybe misunderstanding the chart. So when we say knock out XBC1, it means we're knocking out the dream complex suppression of DNA repair, right? So they should have better UV light or UV damage repair.
For Lin52, yeah. So they should see what's there.
Like if these things are doing at all what we think they're supposed to be doing according to the rest of this paper, DNA repair should be improved in those last two models. But what we see is they lose reproductive capabilities entirely.
Yeah. So what's their explanation in Figure 1 for Figure 1F?
I don't want to keep us on this too long. It's just
no.
I'm very confused by it. It's completely backwards from what I would expect given everything else this paper talks about.
Yeah. Well, let's take a look at what they say. Let's take a look at Figure 1F, buried in this paper here someplace. It's Figure 178. Considering that DREAM represses gene expression in non-dividing cells, we hypothesized that the UV sensitivity in the germline would be unaffected. Lin52 mutant animals laid a comparable amount of eggs to wild-type worms upon UV exposure with similar hatching rates, and the Lin52 mutation did not alleviate the germline hypersensitive worms with mutated XPC1. Therefore, DREAM mutations specifically augment DNA damage resistance of the somatic cells. See, and one sentence behind that is, owing to the phenotypic specificity with mild adverse effects under unperturbed conditions and the strong UV resistance phenotype, we decided to focus mainly on Lin52 mutants to further investigate the role. Okay, so that doesn't seem to answer your question.
so maybe XPC1, the DREAM complex's binding to XPC1 is part of the reproductive. No, but then you would have seen it in the zero UV. We can move on. I'm not sure. That was handled by one sentence in the text. Okay, that's fine. We can move on. I'll just write this off my memory as that was odd.
Yeah. Well, I mean, I've for me the biggest paradox is right over here. Right. I mean, I would expect to see way more lifespan with zero also, and no inhibition of lifespan. So obviously there's some other perturbatory effects happening that are not
probably
positive.
all related to the DREAM complex is big, does lots of things, and in some cases it's maybe makes UV damage worse in germline cells. Maybe or something like that.
Well,
these are big and complex, lots of side effects.
Yeah, makes UV damage worse. I mean, yeah, their argument is that Yeah, well, it does. If it's active and acts as a repressor, then it should make UV damage worse. Yeah. But if you have a Lin52 mutation, yeah, I mean, again, we can argue about this back and forth, but without actually anything quantifiable. It's all qualitative, right? I don't know what the percent contribution is of the dream complex to the expression of XPC1 and so on and so forth, and what. So if you've got that giant chart of everything that the dream complex turns on and off, what are the numbers behind that? I don't know. It's just kind of words, right? So they do go up, but okay. So now they look at the damage itself. So they show that dream complex mutants show enhanced repair of UV-induced DNA lesions and alleviate the UV sensitivity of CSB1 and XPC1 mutant animals. So this is they look at here. So they're doing a dot blot assay, which is basically extracting proteins. So let's see, CPD. So these are these cyclobutane pyrimidine dimers, right? So that's basically the type of damage that happens. That's when you zap the cells with the UV. This is one of the forms of damage that happens. Wild type UV, and then you wait 24 hours, and you basically smush up the worms. And these are in L1 larval stage. So this is L1 larval stage. C and B, they look at basically adult worms, specifically nuclei in their heads, which they mentioned. Why did they look at nuclei in their heads? Because that's where most of the nuclei are. So it's a nuclei-rich region. So this is total DNA. This is an antibody against the cyclobutane pyrimidine dimers. And you're basically comparing the difference of repair. So difference of these dimers, when they were just initially zapped to 24 hours later, right? And the intensity basically corresponds to how much more there is. So if you have this, let's see what we have here, XPA1, is that a DNA repair enzyme? Can't keep track of all the mutants that these things. You need a C. elegans mutant expert online here who could just rattle off all the different mutants. XPA1. I'm going to assume it's another DNA repair enzyme. Where's XPA1 mentioned here? Come on, guys. Paper. Sorry, I'm bouncing around here. Somebody else read this paper. Where's the XPA1? The heck with it. It's a DNA repair enzyme. Okay.
I'm looking at this on a page that doesn't have page numbers, so I can't tell you what page it's on.
Yeah. Okay. So anyway, the bottom line is that if you basically look at these cyclobutane pyrimidine dimers, if you have so DPL1, Lin52 again are components of the dream complex. So you'll expect. So you can see right here. So this is the clearest example here. These two guys right here. You have less of them. You don't really even need to do statistics on that. You can see that this band is much lighter. There's so within 24 hours the repair is increased, so they're gone. So there's less damage, which is good. So they don't just go through development. They actually we got a chat here. Okay, what do we have in our chat?
The XPC1.
Okay. Nucleotide repair is initiated either by the transcription coupled neuroprotein CSB1. Yeah, that's it. Yeah, or by the global genome wide neuroprotein XPC1. Yes. Okay, that reminds me. Yeah. So transcription coupled nucleotide excision repair is primarily found in adults. So it's basically, as the name suggests, you have nucleotide excision repair. So that's one of the repair pathways, and it's only coupled when transcription is taking place. So it's, so the cell isn't really kind of paying attention to the rest of the genome, versus in the germline, and I guess in younger worms early in embryonic development, which in this case is L1 larval stage. Well, that's XPA1. I don't know. XPC1 is both branches then recruit XPA. Okay, so they basically converge on that. So XPA1 kind of covers both the global genome-wide nucleotide excision and repair that's found in the germline, and both of them require converge on XPA1 that assembles the rest of the components. Okay, thanks, Micah. That was a big help. Jogged my memory there. Okay, so the repair capacities go up, as we saw from that blot, and here they look at they count nuclei, and what they do is the nuclei that they're counting are, what is it? So alpha CPD. So basically, they're looking at they're staining for the cyclobutane pyrimidine dimers in adult worm heads. This is DAPI staining, so basically stains for nuclei. This is that antibody against this in the adult worm. So I guess that's the way they do it. I guess you can grind up the whole worm and do the same thing. I'm not sure why it's done this way for this analysis.
More bright dots are better here.
Less is better. Well, less of this. So this is damage. So basically, the green is damage. Green is these cross-linked cyclobutane pyrimidine dimers. So the UV light will basically make these aberrant nucleotides, and then you have an antibody that can actually stain for them. So you can see that. You can see, qualitatively, it looks like the glow goes. There's less intensity if you overlap it with the nuclei, and this is supposed to basically represent that as well. And they're showing that this is going to be a statistically significant drop off after 60 hours of UV
and this
zapping.
Is, okay, so at time zero, they did the zap one time, and then recorded again 60 hours later.
Yes.
And so in the Lin52, we see a decrease between. An idea here is that because DNA repair was more effective, the damage from the initial zapping was repaired over the course of 60 hours. Is that correct?
That is correct.
Okay.
Yeah. So that's kind of, again, I'm not a worm guy here, so I don't know why certain aspects were, certain types of damage were looked at, or why certain assays were done in a certain configuration, but that's why they, that's what they did. That's the data they have. So on to the rest of this figure here. So now they're looking at mutants in, so row D here is, so we have Lin52 again, our mutant that, downregulates the dream complex, right, and upregulates repair. So this CSB1 is a component of transcriptionally coupled nucleotide excision repair. XPC1, as mentioned before, is genome-wide nucleotide excision repair that's found, primarily in the germline. And this is the double mutant, where you basically have both of these knocked out, right? So that should be kind of a much more dramatic effect. And then they have, again, zapping cells with UV light. This one, you can see that these bars look different. So, pay attention here. Obviously, this is the millijoules per square centimeter. I'm not sure why they had, different ones. So here's 0.1530 on E for the, GG NER is 0.4060. It could potentially be that you need a higher UV intensity to see the differences later on. And here it's much lower because these are probably going to be much more severe. So 0.13. Yes.
This feels like I mean, this plus the other things we were discussing earlier, it feels like that XBC1 is activated by UV. Like UV light is doing something to it that's then causing it to then take some action. Because the fact that you're seeing that, just 1, 2, 3, which normally has basically no effect on a wild type. Like that's just such a low dosage that the wild type just ignores it. You add that XBC1, now all of a sudden, like it looks exactly the same.
Well, if we look up, so my memory's a little vague here, but I think mutations in xeroderma pigmentosa in humans, you're very sensitive to UV light. So if somebody could do me a favor and Google it, I think you get like really bad scarring if you go out in the sunlight. I think that's like and you basically have higher elevated rates of skin cancer. I could be wrong about that. So I think it basically plays a big strong role in protecting humans against UV light. So it could very well be upregulated in response to those types of damage that occur.
Yeah, this like I said, just this one combined with those previous charts that we saw, this XBC1 seems to be like when there's UV light present, very significant and noticeable changes that are, not related to damage repair seem to show up. Like failure to reproduce shows up when you have UV light plus XBC1. And in that chart, when you have UV light plus XBC1, you see larval development fail. Even just a little bit of UV light, like enough UV light that shouldn't have had any negative effect whatsoever, which just really makes me feel like the UV light is activating this XBC1 or, causing it to behave in some way that is then maladaptive, or maybe it's some, defense mechanism gone wrong. Whatever it is, it seems like the UV light is, when combined with XBC1, is creating problems, not solving them.
Yeah, perhaps in worms. Yeah, again, we're looking at worms here. So it's definitely more responsive. So the mutation in these genes in humans, right, causes this condition. That's what it's named after, xeroderma pigmentosum. So yeah, I'm gonna stop sharing that. Okay.
No, the XPC is named after that right there. That's what you're referring to.
I believe so. Yeah.
Oh, interesting. Okay. Which definitely does suggest that it is UV activated in some way.
0. Yeah.
Okay.
See.
It's starting to make sense.
Yep. Xeroderma pigmentosum complementation group C. Yeah. XPC1.
Okay. Well, there you go.
Yeah. Okay. Okay. So let's see. Where's my paper here? Whoops. Back to the paper. All right. So what do we see here? Let's see. So Lin52. This is basically a repeat of what we saw before. So it's improving the worms to go past their larval stage compared to wild type, right? So our Lin52 mutations. If you have mutations in CSB1, which is the transcription coupled nucleotide excision repair, or the global nucleotide excision repair, which is XBC1, you have massive defects, and especially with both of them knocked out. And again, this is way lower dosage. So now we're looking at 1 millijoule versus 40 and 15 and 3. And I think what they're showing here is that you don't have any. So if you have a Lin52 mutation, if you have both of these components knocked out, which it basically blocks the repair completely in this case, or prevents progression through the larval stages. Yeah, Micah.
My last comment on this, because I finally clicked in my head, and I just want to make sure anyone who's listening gets the same click that I just got. XPC1 is something that is known to increase the negative effects of UV light. And so it's used as basically a control, and then they're seeing if we have Lin52 plus XPC1, does Lin52 counteract the negative effects of XPC1?
Yeah, so so just realize that XPC1 is a mutant. So this is knocking out that gene function.
Yes, and so we basically created these worms that are UV sensitive.
Yes, correct.
The XPC1 mutants. And then we're seeing, okay, now if we add the Lin52 to a UV sensitive worm, does it have a positive effect?
Yes.
Makes all these charts now suddenly make sense in my head. I get it.
Perfect.
We can move on.
So basically the Lin52 is sort of compensating by upregulating other pathways that'll take over at least partially the role of XPC1 or CSB1 were they present.
Gotcha.
But if you kind of disrupt all of them, then Lin52 has nothing. That mutation has nothing to work on. Yeah. Okay. So all right. So that's basically what they're saying there. So now in figure, I kind of joke when I see figures like this. I'm like, machine is meant to read this, not humans. So they do basically, well, what they do here is they look at the mutations, right? And kind of similar to when we started with this paper here, where they did a promoter analysis and showed that these promoter elements were, that the dream complex overlapped with a lot of DNA damage response, preferentially with DNA damage response genes, higher than just the average, if you look throughout the entire genome. This is basically showing that where's the figure legend? Oh, is that figure 4? I guess that's figure 4. Is that a thing these days to put like figure legends three pages later from the figure?
God, I hope
not. It's even worse when I print out this paper on two sides.
No, man, you have to flip back and forth until you find that.
Yeah, it was like, what? I swear 5 years from now, they're going to also print these figure legends upside down 3 pages later. Not sure why. But anyway, is that the right figure legend? No, that's figure 4. What is this? Figure 3 we're on. Yeah, there it is. Okay, it's before. Okay. So they're looking at basically which genes are enriched, right? So obviously, so the dream complex represses them. If you have mutations in Lin52, and you look at expression, do various expression analyses. Which genes get upregulated, basically, right? Using a variety of techniques, quantitative PCR, RNA-seq analysis. And, the bottom line is that there's a lot of things that are upregulated, and a lot of them fall into this category of, no surprise, DNA repair, cellular response to DNA damage stimulus, double-strand break repair, so on and so forth. What do they look at here? So I think these are, don't want to really kind of belabor this. I think we can spend more time to see what, later what gets enriched, but really the bottom line is that Lin52 versus wild type, et cetera, when you have mutations in these dream components like Lin52, you tend to have upregulation of genes that kind of matches more of what's enriched in the germ line, which is these DNA damage response genes. So that's kind of basically it. I don't know if anybody wants to kind of go through this in more detail and look at these Venn diagrams of what's upregulated, but I think that's really the gist. Okay. So we could certainly, and I'm not sure why they picked this table here, because these are all various DNA repair enzyme components, and they just basically show that see, so Lin35 versus wild type, Lin52 versus, I guess this was the mutant that was screwed up. So which ones are bound by? So fold enrichment, basically, fold change if you have these mutations versus, versus non-mutant, right? So ATM1, ataxia telangiectasia mutated. So these are all mutations that are found in DNA repair enzymes, and you have a higher fold expression throughout a lot of these. And I think there's more in the supplemental data. And I'm not really sure why this table was put front and center for all of these guys. Yeah. So a lot of different DNA repair enzymes are basically upregulated when you have a derepression of the dream complex, right? So, and there's a list. If there's your favorite one in there, you can take a look and see if it goes up. Looks like most of them are not quite twofold, right? So 1.5-fold looks like the average. Some are a little bit more than twofold, but that's, so you get a modest but statistically significant increase throughout all of them. Next, what do they do here? So now they look at basically very similar damage responses, but not just to UV, but now they look at a whole bunch of other, because this dream complex represses a lot of different components of a lot of different repair pathways, and these repair pathways respond to a lot of different genetic insults. So they basically throw a lot of different things on these worms, these poor worms. So ionizing radiation, measured in grays, methylmethane sulfonate, which is a DNA alkylating agent, cisplatin. I'm not sure exactly what mutants, what sort of DNA lesions it induces. It's a chemical mutagen, I believe, that's used in it's a platinum-based mutagen that's used in as a cancer therapeutic. Yeah.
Would one expect the mechanism of the DNA, like the thing that caused the DNA damage, to have some effect? Like if you're damaging the DNA, you're damaging the DNA. Why does it matter if it's UV versus something else, like chemical damage?
Yeah, because because it's different flavors of damage. So let's say one damage is just a double strand break. So if you have ionizing radiation, it'll do a lot of things, but it'll, I think, predominantly break the DNA, so through both strands, and that will activate a different kind of cluster of proteins that will affect that. Either if it's a post-mitotic cell, it'll be non-homology end joining. Actually, hold on a second. Let me.
So the actual damage is theoretically different here between the different modes of damage.
Yes. So, okay. Yeah, so share. So like, for example, here. So one thing that they mention in, I think, the paper is that in post-mitotic cells, you have things are shifted towards this thing called non-homology end joining, which uses this subset of proteins, Q70, Q80. And so this is if you have ionizing radiation, make a double-stranded break, versus another group of proteins which basically do homology-mediated repair that require a template, and I think that's predominantly in the germline. But this is already a difference in proteins depending on whether it's in one type of cell that's post-mitotic or mitotic. This is a little bit more cleaner because it uses a template to make sure that there's no. This one is known also as air-prone repair. So there's a whole bunch of other mutation, other genes that are involved here, and for other mutagens, you don't have a double-stranded break. You have these cyclobutane pyrimidine dimers, which are basically like covalent bonds next to basically nucleotides that are adjacent to each other, and that little area has to be snipped out by one enzyme and then basically patched by other enzymes. And then there's yet other mutagens that basically intercalate or stick into the base pairs, and when you have DNA polymerases come by, they can skip over, or they sort of act as kind of a speed bump, and they'll add in extra nucleotides or remove nucleotides as a consequence. So and so on and so forth. So different mutagens can basically cause all sorts of different types of chemical signatures that are specific for that type of mutation, and it requires different subsets of things. So over the many eons, the cells have basically evolutionarily collected a whole palette of enzymatic pathways that can repair all the different insults that the cells have expected to have come across, in the harsh world. Yeah.
I don't want to get distracted on a tangent. Do we know why germline cells seem to have the better repair mechanism, and why that seems to get lost in somatic cells?
Yeah, well, I would guess because somatic cells, at least for metazoans, are evolutionarily dead end, and if you have germline, if you have mutations in the germline, that's going to propagate to your offspring, and you might actually cause a whole species to collapse and fitness to basically decrease. So yeah.
But is there some disadvantage to it that we know about? Or as far as we know, it's just strictly better to do the germline mechanism, and there's just unfortunate that our somatic cells don't.
That's a good question. I'm not sure if people are in 100%, agreement to this. I think, I think people would say, when people being evolutionary biologists and people in the aging field, that it's just a consequence of selective pressure, right? So if all this repair is happening in the germline, and the germline is propagating, and you have enough species and everything's going well, and damage is accumulating in the somatic cells, but you have enough offspring, then who cares, right? It's just basically it's not even being selected for. We obviously care, but as far as evolution is concerned, it's done its job, right? It's like, what else do you want, right? So, but
I'm thinking about this. I'm thinking about this from an engineering standpoint. Like, is this something where we could take the germline's mechanism and somehow get it into the rest of our cells, and would that just be a strict improvement?
No, well, I think that you're getting right to the heart of the question. That's what's going on. What's going on? Essentially, the question being asked with the OSK factors, right? Can we do what we can we improve what's happening in the germline and other cells that seem to have enhanced repair capacity, and put it into the rest of the cells? I think the answer is sure, why not? But the more complicated answer might be, at least for some of the stuff like this dream complex or ways that you go about it, it could be that the way the system kind of evolved, it sort of ended up being kind of a Rube Goldberg device, right? Where you have to be very engineering wise, where it's basically layered onto other legacy systems, and if you and your simple question, well, can we just do what we do, improve what's going on in the germline, and and will that theoretically basically make the somatic cells live longer and healthier as well? And it's like, sure, but while you're doing it, you're like removing like a Jenga piece, and other things might topple over, right? So if you can do that without perturbing the other systems, then the answer is yeah, sure. I think
so. There's no known downsides, but of course there's many potential unknown downsides because of the Rube Goldberg Jenga machine.
That's my kind of hand wavy explanation. I think
a good explanation.
I think it probably holds. I think we need to quantify. Which how many interactions there are with the Rube Goldberg components. But I think that's the short answer. That yeah, you can do it, but you got to be aware that when I think it's just kind of like probably like software, right? It's like if you have a lot of other craziness happening there, it's like, well, can't we just put a patch here? But then that patch might affect other things, and then you have to really go down that rabbit hole to make sure that that it's not dependent on the fix that you put in that's perturbing something further downstream. And another,
I like to tell people that cellular biology is basically just the worst software ever written. Like, you imagine for software engineers, like what you're describing, this kind of, you touch one thing and something else breaks. That's kind of the worst case scenario for software that you try very hard to avoid. And biology seems to have just leaned into that heavily.
Yeah, well, most of our genome is also derelict viruses and other things that aren't supposed to be expressed, and and the system basically suppressed them, and some of these things might actually have eventually evolved some sort of positive role in some other aspects of just getting rid of that. It might actually perturb something else. And the other thing I want to add to this also is that I don't know off the top of my head if adding in more resources to somatic cells to improve its repair capacity might be detrimental in a more kind of fundamental way for the survival of the germline, meaning that it's that whole model with antagonistic pleiotropy, right? If you Yeah, being healthier, being more fit, or being able to kind of repair damage or certain types of damage is a positive within certain, because the other half of the equation for living systems is the environment, right? So you have to realize that these evolved in an environmental system where you're being chased by things that want to tear you to pieces, right? And you have to outrun these things. If you can live twice as long, and be resilient, but you run half as fast, then it doesn't matter. You're dead, right? So if so, that's the trade-off. If the systems have, so there's this very complex interaction here that equations going back and forth where if you can't outrun, or I just used the outrun, but outcompete in some way something else, and if the system has to evolve something that cuts into your lifespan but doesn't, by basically making you faster, but as a result puts more wear and tear on the systems because it's your metabolism is going into overdrive and so on and so forth. Whatever is going to basically give the next generation a survival advantage, or basically propagate the species, that's what's going to be selected for. So it's not always so living longer and healthier. If that cuts into those survival characteristics in a very aggressive, competitive environment, if that's the trade-off, then the species will never propagate. Now that being said, if you take the species out of the environment and put it in a nice cushy environment where you don't have to worry about that, then everything then all of a sudden we're presented with the dilemma of aging, and then we want to solve that.
Yeah, I think the simplest example of that one is that what you're describing, I think, is things that are energy expensive used to be, that's a big downside. Now energy is basically free for humans, right?
Yeah.
Food is no longer scarce, whereas evolutionarily food is scarce.
Yeah. Well, at least for the lucky subset of us, right?
Sure.
Yeah.
Compared to evolution, humans are doing well on food in aggregate, I should say.
Yeah, humans and our pets. Yeah.
Yes.
Yeah. So I'm not going to kind of belabor this, but basically you're seeing similar effects here, where I believe let's see what we're seeing here. So MMS. So I think these are mutations that are supposed to block Lin52, CKU70. So ionize. Let's go back up here.
In those charts, do we strictly want more white, or do we just want to look more like the wild type? I guess the wild type has a little bit of red. Is that okay? And we want to target that? Yeah.
Well, we want to see. What do we want to see?
Sorry, wild type at 0 damage. Is that our target, or is our target just a pure white bar?
Our target is more so more white bar the better, because that means that things have basically become adult, and more of these, so less red or less orange or less L3 or more of it in so L1 is basically early development, so these things are basically tracking towards the adult, which is towards the white here. So for example, Lin52 here, you have more of them get into adult with 360 gray. When you have CKU70 here, that blocks the effect. So see, it's another DNA repair enzyme. It's downstream. So I think see here. So I think they mentioned
some mutant that has suffers more damage, and then this Lin52 fix it, which in this case it doesn't.
Yes. So basically these are I think let's see, pull which one. So in some of these. I think in some of these, like Pol H1, so you get an improvement here with a Lin52 mutation. So there's other enzymatic pathways that can improve it, and so on and so forth. Cisplatin, I think it's the same thing here. Lin52, yeah. So 1 millimolar cisplatin as well. So they're seeing, so in general, kind of, when you have these mutations in these dream complex, you improve the repair capacities, meaning that the cells can go on to a later larval stage. Now, one question I had is, they have, better egg hatching, and they go into they go all the way to the adult larval stage. I don't know what the consequences are for future worms, right? So it's like, are there any later aberrant problems with de-repressing the dream complex, right? Because there could very well be. So you're showing that these worms are more resilient, but then again, looking back at the earlier data there with the lifespans, they do have a lower lifespan capacity, at least some of the mutants, not the Lin52 one, but some of the other ones that are mutants in the dream complex. So, so it's a little bit complicated, because I think this complex basically hits a lot of things. And last, they jump into, what do they jump into? Human and mice. So they use a mouse model of these. It's a mouse model of Cockayne syndrome, which is an accelerated aging phenotype. It's a repair defect, and these mice are basically, to recapitulate, these mice have a mutation in this enzyme ERCC1, which is a repair enzyme. For human cells, so here what they're doing is they're trying to see if they can inhibit the dream complex using this DYRK1 inhibitor, or these two chemicals, harmine and this one called Indi. And they basically target an upstream regulator of part of the dream complex. So it's what is it? So it affects phosphorylation. Let's go up here. Okay, in mammals, dream components not only form the dream complex, but associate other complexes. So chemical inhibitor of the DYRK1 kinase, which phosphorylates Lin52, which is a component of the dream complex, which basically is required for assembly of the dream complex. So basically, if you inhibit this kinase, which phosphorylates, it basically prevents the assembly of part of this dream complex. So it's basically acting as a negative regulator. So you're adding these chemicals which inhibit this kinase. So instead of making mutants, you're using chemicals to essentially downregulate this dream complex. And then seeing what effect that has on cells. So what are they? Well, actually, A and B is this more. What's this figure? Not the figure. Okay, so they do the same thing. Overlap between DNA repair genes in humans and the genes bound by dream. Okay, so basically they're showing that these dream target genes are also upregulated preferentially when they add these chemical inhibitors of this DRK1, basically activator of the dream complex. So I believe that's probably, so you have an upregulation predominantly of these DNA repair genes, and there's probably somewhere in the supplemental a more extensive list of this change. So what do? They use these human cells, U2OS, I believe. Where is this? It just seems later it's harder and harder for me to find the figure legends. Let's see. Here's where they mention the cells that they use. Come on, figure 5. Where's figure 5? If I publish a journal, I would have like boldface. Figure 5. This is where we start talking about figure 5. This is where we start talking about figure 2.
If you view the paper on the Nature website rather than PDF, they actually just have a nice little thing that you click on a figure and it will show you its caption right underneath it. And you can. That's
what I need to do next time. So they use
the PDF version is not great.
Well, it's not even great printed out. As the dream complex represses gene expression in G0 cells, so G0 is basically quiescent non-dividing cells. They basically they used for this assay serum star confluent U2OS cells to obtain quiescent cell populations. So they use this one cell type. Let me stop sharing. What's a U2OS cell? I know we talked about them in the previous cell. U2OS cells. Epithelial cells derived from a sarcoma tibia of a human a long time ago. It's a cell line. So it's a cancer cell line with epithelial morphology. So epithelial cells that, skin and line other, the outer surface of tubules and whatnot. It's derived from the bone tissue. I'm not sure why they picked this particular cell line to do these assays, to be honest. You could pick a lot of other cell lines. So I'm not sure why U2OS cells were used. Yeah, I'll just leave it at that. They don't really mention that. Okay, could be some reason, and somebody knows off the top of their head why U2OS cells were used, and not a panel of. You could also use a panel of other cells as a comparison, but but these are cancer cell lines, so just keep that in mind that these aren't normal human cells. They are human cells, but they're not normal health. They're not primary cells, right? That are been biopsied from somebody and aren't cancerous. So what they show here is that harmine and indi are repressors of the dream complex. This is a Bax scatter plot, so fluorescence activated cell sort, and they're looking at components annexin 5. I think it's all annexin 5. So these are basically these are markers for apoptosis. So the cells are basically, you zap them with UV, the cells undergo apoptosis. You block this pathway. Supposedly the cells are doing repair, right? But maybe they're not, because what you're basically showing is less apoptotic cells. So if you do control UV, here's the percentage of apoptotic cells. So you're looking at this box right here in the upper right-hand quadrant. There should be less of them. So you can see here, annexin 5, 7 AAD, that's another marker. So two markers. I think there's two markers of apoptosis. So if you look at, this quadrant here, it should go less. So less apoptosis when you use these inhibitors, right? And this is, so you can even see here that it looks less. So all these dots is basically a cell that's lit up and is basically indicating that it's undergoing apoptosis, meaning that it's undergoing a programmed cell death pathway, which happens when repair usually can't happen properly, right? So then that's sort of the default mechanism that the cells die. So I guess, the amount of UV given is enough to kind of, so I guess these U2OS cells don't have the apoptotic pathway inhibited, which is a hallmark of many cancers. But I guess maybe that's one reason why the U2OS cells were chosen, so that they're capable of undergoing apoptosis. So control UV, more cells are apoptotic. Harmine, they go down. Same thing with indi. What do we see in H? A, B, C, D, E. Oh, that's just F and G. What are we doing in F and G? Percentage apoptotic cells. Oh, methylmethane sulfonate. So different mutagen. So less cells are undergoing apoptosis, but the assumption here is that repair capacities are improved. But obviously you need to test that, because these are aberrant cells. They're cancerous. So they could still be just not going through apoptosis and having increased aneuploidy and whatnot, and other aberrant problems later. So I think we still need to check to make sure that repair is actually taking place, even though you've blocked apoptosis from happening here. And okay, so the last thing they do is they look at mice. So this is in vivo. So these are mice with these ERCC1 mutations that are, I believe, replicate Cockayne syndrome, which is,
Is there a reason that they didn't just do one of the other things like UV exposure or something to the wild type mice? Like why they used the mutant mouse?
I'm not sure. I think they were kind of leaning more towards I think they were leaning towards trying to show that you have an improvement of functionality here, or an improvement in repair in a mouse model that mimics a condition that is considered to be an accelerated aging phenotype.
I see.
For that reason, and that they could basically claim that this improvement can potentially alleviate, it's progeroid-like conditions. Of course, not all progeroid-like conditions are similar.
Right.
Hutchinson-Gilford is a different protein that affects nuclear stability and how chromosomes are arranged in the nucleus. And it could very well be that A, this checks off the box for a progeroid condition, and B, they had access to these mice in a facility where they were basically working. So here they're basically doing a similar effect, similar assay up here, which they're using a tunnel assay, which looks for, which marks cells that are undergoing apoptotic cells, basically. So tunnel positive cells. So if you have, this is outer nuclear layer. So they're looking at so these are cross-section through eyes. So this is basically, I think these mutations, this ERCC1 primarily affects, for whatever reason, so eyes. So mice mutations in ERCC1, which encodes a nerve component, lead to strong progeroid phenotype and premature death. Analyzed loss of photoreceptor cells in the retina of ERCC1 double negative mice, a hallmark of Cockayne syndrome that can also be observed in nerve deficient mice, which is basically a human condition, by performing tunnel staining of the outer nuclear layer of the retina. And then they also basically were looking at gamma H2AX signals, so staining of this histone, which is indicative of damage. So the bottom line is that when they looked at these areas of the retina plus harmine treatment, right, or untreated, so that's supposed to basically block the dream complex, you get less tunnel positive cells in these ERCC1 mice. Also, well, they say it's not statistically significant, but okay, well, you could look at the graph yourself there. It's a wild type plus harmine. Maybe I think this is only like 3 or 4 mice, so it's not a lot of mice. Or maybe it's 7 mice. It's not a huge amount versus the worms, which you have like over 1,000 worms. So you have less apoptotic cells and less indication of DNA damage when you look at another cell layer in the retina when you have plus harmine. So they mentioned that this potentially could also act. This could be used as a therapeutic in their discussion for these various conditions by basically affecting the dream complex. That being said, I think more work needs to be done because it's a very complex, no pun intended. There's a lot of downstream effects that potentially might happen. One thing that I'd be worried about is, cells going forth, obviously, not undergoing apoptosis, but replicating because you block this complex. But did you fully upregulate all the repair capabilities, or are you nudging some of the cells into kind of a more cancer phenotype? That's would be my caution there. DNA repair is essential for maintaining the integrity, obviously, of genomes, both in germline and somatic cells. I'm going to do a little bit of digging and see what other types of analyses were done in C. elegans to see if upregulating certain pathways, or a variety of different pathways using other techniques is capable of increasing kind of basal lifespan without the need to disrupt the genome using a variety of insults such as ultraviolet light, so on and so forth. Because really that's kind of what we're after, right? We're in an environment where we're not being subjected to all of these conditions, but we want to basically extend lifespan beyond the low basal rate of genotoxic stress that we're experiencing right now in an optimized environment. So we want to kind of get the somatic cells living longer and healthier, beyond the basal rate of genotoxic stress that's already low level, that's happening right now, that's intrinsic, that our cells are generating as they basically metabolize and proteins misfold and accumulate and other types of damage basically accumulates over time. Yeah. So
this dream complex, it suppresses expression of certain genes, right? And
yeah.
Do we know if, and this may be a very basic question with a simple answer or a very complicated one, or I don't know, but do we know if you were to introduce another gene, such as maybe separate from the genome or somewhere else in the genome, would the gene complex also suppress that, or is it specific to the location within the genome that it gets suppressed?
I would assume that it, as long as it's in an area where it's accessible transcriptionally, I would assume that if you put it someplace, if you put it in some other location, that the dream complex should be able to repress it as well. Yeah.
Okay. Just trying to think of other ways that we could improve DNA repair by getting upregulation of those things we want without having to try to mess with this really complicated dream complex thing. Such as maybe some other way to get those proteins.
Well, the more basic way to do it, the more traditional way to do it, and is overexpression, right? You put in an extra copy of these repair enzymes into an animal, either transiently or stably within a particular region. And then an even more kind of subtle way to do it is you put in one copy or two copies, and then you have an inducible promoter where you can basically throttle the levels of basically the particular components, because too much could also be a bad thing. You might actually end up disrupting some complex. So it's just like basically throttling the level of, like we spoke earlier, of the OSK factors. So you could potentially do the same thing with the repair enzymes as well. The same basically concept, and that might be a more subtle way to do it. So this has piqued my interest and curiosity to kind of maybe go do some literature searching through the C. elegans papers to see if these types of experiments were done in this direction. Certainly a lot of other organisms, yeast, for example, improving DNA repair capabilities, I think, is known to improve chronological lifespan, so on and so forth. So definitely lifespan is enhanced, basal, without requiring to add in excessive amounts of other types of exogenous genomic, damaging agents that, over and above what your cells are already kind of churning out. So I think really that's the direction that we should be going, is seeing if we can, improve those capabilities, that are already kind of evolved to whatever their optimal or suboptimal level is during normal metabolism without any external agents being added, right? Can we enhance that even further?
Yeah, I think the other thing that I already brought up, and just restate here at the end, I think that I would really like to see if this dream complex, if its expression changes with age. Anything that's trying to solve aging, I feel like, kind of, for me, the base thing to get me interested is, does this thing change with age, or is it something fixed over a lifetime? If it's fixed over a lifetime, my interest is very low. Whereas if it's something that increases or decreases with age, then I'm very interested in, okay, how can we make this go back? So if it's like when you're young, the dream complex is, very rarely expressed. And when you get old, it's expressed very highly. Then it definitely feels like something, okay, well, how can we get that expression turned down?
Yeah, I'm basically just started going through PubMed right now while you're talking, looking up exactly that to see. Nothing is really pop. Oh, sorry, I just misspelled complex. Obviously it's important in cell cycle regulation. What did they mention? Yeah, they mentioned this in the end of the paper here. Let's see, so they mentioned intriguingly indirectly, right? So intriguingly, DYRK1 kinase is overexpressed, meaning that the dream complex is upregulated, or the activity is upregulated in Down syndrome and involved in neurodegeneration in individuals with this condition, as well as in people with Alzheimer's, Parkinson's, or Pick's disease. I'm not sure what Pick's disease is. And it says it will be interesting to explore whether dream complex inhibition could prevent DNA damage-driven conditions such as stem cell exhaustion, neurodegeneration, and premature aging. But they don't mention what you just mentioned there, which is is it perturbed in people that are normally aged, and
right.
and do supercentenarians have de-repressed dream, or less of it, yeah, right? Repressed dream complex activity. I don't know. Or there are particular mutations that cluster in the dream complex in supercentenarians, right? Is this one of?
Yeah.
There could be there could be, that information could be there. It could be latent in some database. If people have sequenced supercentenarians and there might be some mutants that overlap with those components, or not just the dream complex, but again, things that regulate the dream complex as well. So we're looking at kind of expanding that to the dream complex outwards. So yeah, those are good questions, Micah. I don't know. Those are very basic, good questions, and could very well be that the answer is as yet unknown, or could just be that I haven't. I just need to do some more proper search term plugging in to PubMed and other databases to see if somebody has done an experiment in that direction.
Yeah.
But a cursory search hasn't pulled up anything for me. So yeah.
Anyone listening want to go answer a good question? Go answer that one.
Yeah, that's number one question right there, right? And if the answer is unknown, well, there you go. We have another series of experiments to perform.
That's right.
So yeah, so that's as always, we not only cover the papers here on our journal clubs, but we also offer potential future experiments to anyone listening.
I actually went into this journal club meeting very I was like a big fan of this dream complex downregulation, but after just this discussion and talking it over, I'm now much more cautious. I think I'm very convinced by your claim that this is a really big complex that probably does 1,000 different things, and messing with it is likely to have side effects.
Yeah, and there's subcomponents to it, so it could very well be, not all mutations are. I don't know what these particular mutations do or how severe they are, but there's mutations that basically are weaker. So it could be that you have a mutation which slightly skews the dream complex to de-repress. Maybe there's mutations that cause the dream complex to de-repress certain, a big chunk of certain DNA repair enzymes without affecting other, other things that deal with the cell cycle regulation, or so it could very well be that there are subtle mutations that could have a really positive and powerful effect. And it could be that the way we find these mutations is going through databases where you do a mutational analysis on humans and also model organisms that live longer, or maybe our magical AI overlord systems can basically help us and zoom in on those mutations and tell us where to look. right, without screwing up other pathways as a consequence. What, Steve? You disapprove of our AI overlords?
Stop using AI as a get out of jail card here. Everybody does this, and it doesn't work.
It's the new, what is it, Deus Ex Machina, right? Where you basically the plot element in a movie is that something happens out of the blue and saves everybody, right? It's like,
yep.
It is. It's a plot hammer, as we call it.
Gentlemen, we will use AI to find these mutants.
Yeah, I mean, AI is obviously a useful tool, but it is just a tool, and it does have its limitations. So yeah, I do hear this oft repeated. It's a pet peeve of mine in like the community, the transhumanist community especially. They go, oh, aging. Oh yeah, don't worry, but AI. No, there's no but AI. It's just a tool, dudes. It's just a tool. It's a useful tool, but it isn't like the complete answer to it. So that's why I'm giving you the head shake of disapproval there.
Well, I use it as, tongue in cheek, because I don't know anything about it. I don't code these systems. So I just that's my ultimate hand wave right now. So people used to use other things to kind of hand wave things away, and now AI is becoming the next kind of hand wavy sense.
Well, I just spoke to Irina Conboy last week, and whilst we thought AI was interesting, she still believes that human collective consciousness still trumps AI, which, yeah, I'm inclined to agree. If enough people get together, we can do like amazing things that supersede AI. At this moment, God, there's going to be some fanboy rage in a minute on me.
No, this is a great experiment. So that game that was around years ago called Fold It, where people basically solved certain protein structures, and they basically, it was sort of like some certain people were very good at it, but also it sort of aggregated everybody's abilities to do so. So it'd be interesting to find out because right now they're using AI systems to basically do protein folding, and supposedly they're extremely successful. But I'm just wondering if we have a comparable system in place where basically you open it up to a billion people, right? So it's unfair to compare one human to like an AI system, but maybe a fair comparison is the collective brain capacity of a billion people basically working on a problem. Each individual as like a super neuron, right? Compared to like an AI system. And I think the threshold of where AI might become really superior is obviously if our collective capabilities are finally beaten by like some machine system. So yeah, like playing like having Deep Blue beat like a grandmaster, I think is unfair. It should be like Deep Blue playing like 100 or 1,000. Yes, exactly. Simultaneously, where they're all basically like, they all basically look at a move and they all basically decide what the best move is. And then you basically aggregate that and pick the best move from all of those grandmasters and then pitch that against the machine and see what happens.
That'd be a really interesting game to watch. I would watch that.
Well, when you think about it, journal clubs like this are also a collective form of intelligence as well.
Yeah.
So just putting that out there, I think why not both? Use AI for what it's good at, play to its strengths, and humans play to their own strengths. So I think there's a case for us both.
Exactly. I think you summed that up very well, Steve. I think everybody here on the panel contributes a lot. Thanks for those insights, Micah, and everybody else who's participated and John, Michael, et cetera, everybody in the past, because it's not just me, but everybody else comes up with great ideas, which we then can act upon, right? So this is a collective endeavor to go through the data and come up with novel experiments and good questions that ought to be answered.
That is one of the purposes of the Journal Club. So, mission successful. So watch out, AI. We're coming for your job.
Yeah, I guess.
Maybe. Although I'm of the mind that if we can automate it and it works in a satisfactory manner, we should do it. Like Arthur C. Clarke said, automation is designed to give us more time to play. I'm paraphrasing here, but that's basically what he said. So yes.
I want a food replicator.
A food replicator, yeah. I'll the first thing I'll order from it is Earl Grey hot.
I want to put
this is.
Yes, it's a Star Trek reference. Yes,
slight change of topic. I always assumed that Steve and Fatima were related due to the last name, but one of you is in the very far north right now, and one of you appears to be in the tropics. And so I'm very confused.
No, this is my back garden.
Yeah, you're sitting on a beach, and Fatima appears to be sitting in a very cold climate, very far north. Those are some crazy northern lights.
Well, the UK has some extreme microclimates, so this is so if you face south in Steve's backyard, that's what you see. If you face north, you see that's what you see. It's really wild. Yeah,
I see. Okay, makes sense now. I get it.
Yeah, it's just wild here. So, but no, I'm actually sitting in the conservatory where we've got a palm tree and a pond with a wave machine in it and seagulls. Anyway, so, or it could be a screen, an animated screen background, maybe.
I refuse to believe.
Are you just hand waving that it's AI, Steve? Is that what you're trying to tell us?
Yeah. Oh yeah. It's just AI.
Actually, the background is real. Steve isn't. He's generated.
I'm generated, and
it's the other way around.
I'm actually responding in a way that is most likely to correspond with what you've said. I am really a program.
Yeah, the palm tree is real.
I'm still more realistic than Huawei's AI AI avatar, which is not really a thing at all. So that was hilarious. They did a tech demo for Huawei, the phone manufacturer from China, and they were trying to pass off that they got this AI, and it was like an avatar of a woman on the screen, and it was so obvious that all it was this was in Thailand, and it was so obvious that it was just basically a big screen with somebody sat behind it talking to the person talking to them at the screen, and it was like they had a Thai accent, for goodness sakes. It was like ridiculous.
So this is the modern mechanical Turk. You know what I'm referring to?
It's rubbish, man. It's like this Huawei AI. They said, "How old are you?" And it was like, "Yes, I am 27. I am young and beautiful." It was like ridiculous. It was like, it's so obvious that it was just literally somebody speaking. It was like what we call booth babes talking into a microphone. It was ridiculous. I mean, AI.
If done correctly, like Max Headroom, it could be amusing.
Yeah, that was funny, actually. Yeah, it was funny. That was. No one really knew what it was about, but it was funny. I still don't know what it's about 30 years later. It was weird. Yeah. Anyway, before we go down the AI rabbit hole, which we kind of already have.
We have something coming up later at the summer, right? Don't we? Something.
Yeah, but I ain't really got much to say about it because they're just giving me crumbs. So, I can't say much about the conference beyond it's happening. Was that you?
Sorry, that was a train.
A train. Oh,
yeah.
I've got a palm beach in my back garden, and you've got a train.
That's
fair
right.
enough. Yeah, so we're having a conference. There isn't really a lot to say about it. It's going to be cool. If you've been to the previous 6 annual conferences, you'll know what to expect. Lots of speakers. are going to be announced soon. We haven't announced any, so I'm hoping the events team get their skates on. So I've actually got something to talk about next time. I do know some of the guests that are yeses, but I'm not allowed to say anything. It's going to be cool, guys. So if you want to keep up, find out what the announcements as they happen, just visit us at lifespan.io/EARD or /conference. That works as well. I know it does because I made sure it did last week. You can find out more there, and if you'd like, you can also sign up to the newsletter so that you don't miss any discounts or any news about that conference. But it's going to be cool, guys. It's also going to be virtual and physical in New York, so there'll be a variety of ways to get involved. I'm just going to say that it's going to be awesome, wicked cool, and hope that I have something more to say about it next time. Thanks, marketing. And that's it. So I guess we will be back next time around this sort of time of the month with some exciting paper to talk about. Thanks to everybody who's made that possible, the Lifespan heroes who are here and who are also not here, but help support this and the other things that we do. So thanks for that. And Oliver, anything to add? Any closing thoughts?
No, we will, as always, pick something very relevant and topical, and yeah, I'll see what's out there in the pipeline. I think I saw a tweet recently from David Sinclair about OSK factors and in primates. So I gotta take a look what that's all about. So always excitement in that field. But then again, there might be something else that comes up super exciting next week. So we will find something relevant and exciting. And if anybody has anything that they want to discuss, please email me, and we'll take a look at those papers.
Yeah, we do requests.
Is that primate paper actually out, or is it just a teaser about a paper that will be out at some point in the future?
Right now, I just saw a tweet, so I'm not sure. I think
I saw the tweet as well. I think I saw the tweet as well, and I think that was one that they said the paper will be out in like August or something. It's like a teaser for a paper that's.
I'm pretty sure I saw a preprint.
Okay.
Earlier today, I didn't have time to stop and read it, but I know it was from Sinclair and that, so they may have actually done it on that preprint server that no one knows the name of.
Okay.
You know, the one that everybody just calls BioOrcs. I'm sure it's not.
I think it's a bizarre way to spell bioarchive, but I could be wrong. Yeah. It could be
It's just BioOrcs. You know, BioOrcs. Why not? I mean, everybody likes orcs, right?
Anyway, we'll find something relevant.
Yeah, we will. So, adios, I guess. Happy trails, everybody.
Enjoy. So next time we meet, it will be in late May. It'll be much warmer. It'll be tropical climate.
Unless you're in the southern hemisphere.
Is anybody here in the southern hemisphere?
I don't know.
Almost.
If you are welcome to attend as well, even though your hours are backwards and you're probably standing on your head, but that's okay.
And there could be thousands of Southern Hemisphere people watching on Zoom. I mean, on Facebook.
Yeah.
That's true. That's true.
It is true. So, yes, for them, winter is coming. For us, summer is on the way. So,
so yeah, happy summer and Merry Christmas to all of our listeners.
A little early, mate. See you all next time.
Take care.
Yeah, see you guys later. Thank you.
See ya.